Leading a startup team, whether you're delivering a sugar rush, stocking coffee, or getting a regular delivery of snacks, Office Depot has solutions that fit every startup culture, from getting those first business cards and stationery to ordering fleece pullovers with your new logo. To learn how Office Depot and the California Technology Council have partnered to bring you savings on all of these startup essentials and more, go to californiatechnology.org/member benefits. I'm Daniel Levine, and this is the Bio Report. Mesenchymal stem cells may be a powerful way to treat a range of medical conditions, but realizing the therapeutic potential of these cells has been hampered in part by the challenges of manufacturing a consistent product at scale. Cynata Therapeutics believes its platform technology addresses these challenges. The company's lead cell therapy candidate is being developed to treat graft-versus-host disease, a potentially deadly response to the transplantation of cells from a donor. We spoke to Ross MacDonald, CEO of Cynata, about the company's platform technology, the potential for these therapies, and the indications the company is pursuing. Ross, thanks for joining us.
It's a pleasure, Danny. Nice to chat.
We're going to talk about host versus graft disease, Cynata, and its stem cell platform for developing cell therapies on a commercial scale with consistency. You're working with mesenchymal stem cells. Perhaps we can start there. What makes these compelling for therapeutic use?
Well, mesenchymal stem cells or mesenchymal stromal cells have been under the microscope for a therapeutic application for over a decade now, and there is an enormous amount of interest, not only in their general biological properties, but more specifically how those properties can be translated to therapeutic use. And as data has been collected during that time, both from the academic environment as well as the corporate environment, it's becoming increasingly clear that therapeutically this is a very, very powerful type of cell whose biology can now be harnessed in a wide range of therapeutic application. Essentially, their activity is focused around an immunomodulatory capacity. And what that means in simple terms is that they have the ability to modulate the immune response to allow the body's own repair and regenerative systems to overcome the forces that are causing degeneration and damage. So basically everything in life and in our bodies is a balance. And so what we're trying to do with MSC therapies is tip the balance back in favor of healthy tissue growth and tissue regeneration in response to disease.
What's the range of conditions these cells might have benefit in treating?
Well, there's more than 900 clinical studies underway around the world using MSC. So as you can probably guess, there's a large number of different disease targets that are the subject of those investigations, ranging from the very serious and very widespread diseases that face our societies and economies today, like heart disease and stroke, all the way through to perhaps some of the more obscure diseases, such as, for instance, baldness. They're being investigated in baldness and a range of other conditions as well that represent less, perhaps commercially interesting, but nonetheless biologically fascinating applications of MSC. So definitely the big targets are under investigation as well as some smaller ones. And in fact, our own program in graft versus host disease is a relatively small economic target, a big unmet medical need, but nonetheless not a great commercial opportunity. But it does allow us to move forward into other clinical targets, which of course represent much more attractive economic opportunities for the company and our shareholders.
I know that one of the challenges that companies hoping to commercialize these cells as therapies faced was that they had to isolate them from donated tissue. What were the consequences of that?
Well, you really hit the nail on the head, but the challenge in manufacturing these cell-based therapies is very significant. But it's a challenge that technology faces in many, many different industries. And that is the perception and identification of a market need or a market opportunity and the ability of technology to successfully address that. And you know, for instance, we think back to when man first wanted to explore the moon, and the first step, of course, was to build a vessel, spaceship, that would ensure that astronauts could get to the moon and then back again safely. And so we face the same thing with the cell therapy opportunity, and that is an ability to manufacture sufficient quantities of cells for industrialization, as well as to ensure we have a robust and safe product. And so the approach that has been successful so far to investigate the biology of MSCs has been, as you pointed out, to isolate the cells from donor-derived tissue. And that's typically a bone marrow aspirate or adipose tissue are the primary sources. There are other tissues as well that can be used. But because these cells are so scarce in those tissues, not only do you have to have a very effective and efficient purification process, you also have to cause the isolated cells that you've derived from that tissue to multiply, to expand. And that for cell culture scientists is usually not a big hurdle, but we are talking vast numbers here, and it becomes, you know, a straight mathematical equation. Typically 20,000 or so MSCs might be isolated from one bone marrow donor or one bone marrow donation. But a typical dose of an MSC therapeutic product is around 100 million cells per patient. So the numbers start to become staggering. And so, you know, one has to go from 20,000 to 100 million just to manufacture one dose, let alone be thousands or hundreds of thousands or potentially even millions of doses that are required to satisfy worldwide demand for a major disease target. And that just becomes unfeasible. It's a little bit, you know, to use another analogy, it's a little bit like, you know, discovering gold underneath the White House. I mean, it's kind of so what? You know, you're not going to be able to do anything about it. And our technology provides a second-generation manufacturing solution that is far more industrializable than the approach for that has been used so far, and that is to use donors as the source of material.
You've developed a platform for generating cell-based therapies. What is it and how does it work?
We use a starting material called an iPSC. That's an induced pluripotent stem cell, the discovery which resulted in a Nobel Prize being awarded to the inventor, Professor Shinya Yamanaka from Japan back in 2012. But in parallel, but just a few days behind, were the scientists at the University of Wisconsin, including Professor James Thompson, who are among the inventors of our technology. And the iPSC approach allows us to completely eliminate the need for multiple donors. Of course, being a living cell product, there has to be a human donor at some point. But in the case of iPSCs, that's one donor, one time. Now, iPSCs have a range of important and unique properties. Many of those properties render iPSCs very similar to embryonic stem cells, or ES cells as they're known in the science. And, of course, embryonic stem cells are derived from embryos, and they have been the mainstay of research and, in some cases, product development in the cell therapy area for more than 15 years. And however, they're associated with considerable controversy because they involve the destruction of an embryo to produce them. And in many religious beliefs, that's an abhorrent act. And so with that background of controversy, obviously ES cells represent a challenging source of material, whereas iPSCs do not have that controversial element, but have all of the useful properties that ES cells have, one of which is an ability to reproduce themselves essentially infinitely, in other words, forever, meaning that one can manufacture a very, very large quantity of starting material for the finished product from an iPS cell line. And that means, in our case, that we have a consistent and reproducible product that is always exactly the same every time the product is manufactured. And this is a phenomenon that is being exploited not only by Cynata in the production of therapeutic MSCs, but also by many other companies that have recognized the unique properties and power of iPSCs for the derivation of their own particular type of cell therapy product. For example, companies such as BlueRock, which are using iPSCs to manufacture a certain type of neuronal cell, which can be used potentially to treat Parkinson's disease. So the discovery of iPSCs not only has provided the catalyst for our technology and our company, but also many, many others. And we're now seeing iPSCs being investigated in a wide variety of potential therapeutic applications, not just in our case with MSCs.
Was there great variability in the quality of cells produced from donors?
Yes, every donor is different. It's an unassailable phenomenon of being individuals. Some people have blue eyes, some people have brown eyes, some people are tall, some people are short. And everybody's MSCs are different as well. And that's well established in the literature that MSCs isolated from different donors have different properties. And in fact, MSCs isolated from the same donor at different times, so, week 2, 3, 4 weeks apart, often have very different properties as well, because during that time, perhaps the individual has been exposed to an infectious agent, and their body is responding to that. So what that means is that MSCs isolated from different donors are different. And in the eyes of regulatory agencies around the world, we simply can't have that inconsistency in a pharmaceutical product. I mean, one of the cardinal rules of the pharmaceutical industry is that the ultimate commercial product has to be consistent, so that every time the patient, the doctor, the caregiver uses the product, they can have an expected and reproducible outcome. Obviously, and that's impossible if we're dealing with different MSCs.
Well, let's talk about graft-versus-host disease, the indication for your lead clinical candidate. What is it? How common is it? And how serious is it? We
chose graft-versus-host disease as the first clinical indication for testing our Cymerus MSC technology in human beings. And as you would be well aware, that was preceded by a range of preclinical studies which confirmed that our product was likely to be safe and effective in humans. So that allowed us to move forward into a phase 1 clinical study in the United Kingdom in patients who had graft-versus-host disease. Now, that's a bit of a mouthful, but it's a condition that arises in patients typically who have been diagnosed with some form of leukemia and as part of their treatment require a bone marrow transplant. And in many instances, those transplants proceed without a hitch, and the patient proceeds very, very well following that. However, in a proportion of patients, and it is fortunately a rare condition, in a proportion of patients, the bone marrow is not perfectly matched or effectively matched with the patient, the recipient. And unfortunately, in those cases, we end up with a condition called graft-versus-host disease, or GVHD for short. And essentially what is happening, as the name of that condition implies, is that the donor bone marrow, which represents basically the entire immune system, the donor's bone marrow reacts against the patient, whose immune system by this time, of course, has been wiped out. And it's basically a recognition of, I'm not in the right place, this is foreign tissue all around me, I'm going to react against it. And that reaction can be very, very severe, resulting ultimately in the death of the patient. In some patients, treatment with corticosteroids is effective, and the condition remits sufficiently for the patient to recover. However, a significant proportion of those patients end up with what's called steroid resistance, GVHD, which basically means that there is no other medical option for treating the condition. So these are, as I said, a very, very significant unmet medical need. And ultimately, the outcome is fatal, typically within a couple of years. And it's a progressive disease. It just gets worse and worse, and the patient's life during that time is very, 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 very miserable. So we commenced a clinical study, as I mentioned, in 15 patients with this condition for whom there was essentially no other option. And the primary goal of this study, as it is with any phase 1, is to investigate the safety of the product, and we're very pleased to see that there were no safety concerns whatsoever, and that secondary endpoints, which of course look at the efficacy of the product, were extremely successful. And we, in short, we saw a complete response in over half of the patients, that is, complete resolution of symptoms. Bearing in mind these patients are very, very sick when they come into the trial, typically in a critical care or even in an intensive care setting. And to have a complete response or complete recovery is really outstanding. We saw more than 87% of the patients were still alive at the conclusion of the study. One patient died of pneumonia, which is not unusual in patients with GVHD, and one patient withdrew from the study. So we weren't able to follow those two patients, of course, and that, of course, resulted in less than 100% overall survival. But otherwise, we saw nearly 90% of the patients still alive at the conclusion of the study. So, in short, this study confirmed the safety of our product and also gave a very, very clear indication of efficacy, allowing the company then to move forward to further clinical studies, not only in GVHD, but in other areas as well.
How well understood is the mechanism of action?
And this, of course, is the subject of an enormous amount of investigation around the world, is to try and understand exactly how these cells work. And one of the challenges with cell therapies is exactly that unlike a drug, which you can kind of, to a large extent, pinpoint exactly how it's working, a living cell, of course, is at any one point in time may be working slightly differently than it works the day before or that it works in a couple of days' time, because it's responding to its environment as cells do, particularly cells of the immune system, where they react to the circumstances in which they find themselves by interacting with their neighboring cells and with distant cells by the use of chemical messengers like cytokines, as they're called, and other processes. So the investigation on the mechanism of action of MSCs has focused on how they do communicate with their neighbours. And one thing has become increasingly apparent with that research is that MSCs don't integrate into the patient's cell or tissues, and they, in other words, they don't engraft. Unlike, for example, if a patient has a heart transplant or a kidney transplant, of course, that heart, a transplanted heart, then becomes part of the patient's organ system. In the case of MSCs, that certainly doesn't appear to be the case. They basically have a hit-and-run mechanism, which seems to be centered around the chemical messengers that the MSCs manufacture with their neighbours, which signals to their neighbours either to calm down the inflammatory response or to start regenerating healthy new tissue. And this is, of course, the basis of tissue regeneration and growth in the body in normal circumstances. And that is the response to various signals and messages that occur in our body to turn various processes on or off, as the case may be. So MSCs produce a wide variety of immunomodulatory cytokines. That's well known. They produce little packets of chemical messengers called exosomes that circulate through the blood system and interact with distant tissues. So there's a lot of information about the mechanism of action, but no one really yet has pinpointed exactly how it is that they work. And that's led to almost a kind of a flippant phrase out there at the moment that MSC actually stands for medicine secreting cell, which is quite an accurate description, but obviously it's not intended to be a serious one.
What's the clinical path forward?
For Cynata, the clinical path forward is to continue our GVHD program, and we have a collaboration with a Japanese company called Fujifilm that is very, very active in the cell therapy and regenerative medicine space, as many Japanese pharmaceutical and industrial companies are. So we will go forward to phase 2 in that collaboration, assuming it all goes to plan. And then in Japan, at least, if we undertake a phase 2 clinical trial, we have the opportunity at that point to seek marketing authorization. There's a fast-track approval system in the Japanese regulatory environment that allows cell therapy products to come to market, to be launched at the end of a successful phase 2, which really is a profound change to the regulatory framework for this type of product, but reflects the Japanese government's foresightedness when it comes to bringing new and effective therapies to market, particularly for chronic diseases of aging, such as osteoarthritis and heart disease, stroke, and so on, that represent major economic challenges for Japan as well as many other developed nations. So that's the GVHD program. We also anticipate commencing a phase 2 clinical study in osteoarthritis within the next 12 months. This will be one of the largest MSC studies ever undertaken, 450 or so patients with osteoarthritis in Australia, and that's being supported by the Australian government's peak research funding body, NHMRC. So a very great opportunity for Cynata because, of course, for us to conduct such a study, it would be extremely expensive. And then the third program that we are moving forward with is in a condition called critical limb ischemia, or CLI, which is an area that's received quite a bit of attention for MSC-based therapeutics. It's a condition that typically arises in obese and inactive people who end up with very, very poor circulation to their extremities, particularly their legs, and that results in their legs at its sort of most mild form, a bit of swelling in the legs, and at its most severe form, severe ulcers and ultimately amputation of the lower limb. And this, of course, represents a big unmet medical need as well. A very challenging condition, particularly in societies that, basically are embracing too much junk food and high sugar drinks and so on, that leads to this condition typically through type 2 diabetes as well. So in all, 3 phase 2 programs are on our radar at the moment, and we have a range of very successful preclinical studies completed now in other conditions such as cardiovascular disease, in pulmonary disease and other areas as well, which provide a kind of very good database for the company to go forward either alone or through partnership in other areas as well.
As you look at these other indications, does the therapy differ from indication to indication, or are you using the same cells prepared in the same way?
Yeah, the therapy does differ. For example, in the case of GVHD, the product was administered via an intravenous infusion, whereas for critical limb ischemia, the product will be delivered in the trial, at least, through intramuscular injection. So quite a different approach to delivering the product, and a different approach also to assessing the outcome as well.
You have a collaboration with Fujifilm around graft versus host disease. They were supposed to be at a decision point on licensing the therapy after the completion of the phase 1 study. That decision has been delayed by 6 months. Why is that?
Well, yes, that's right. They were due to have exercised their option in April of this year, and we were certainly looking forward to that proceeding because the basis on which they were making their decision to proceed was, of course, the completion of the clinical study, which, as I mentioned before, was a very, very successful study. So there were no issues whatsoever with the trial. Ultimately, however, Fujifilm just needed more time to complete their review and to finalize the definitive license agreement. And rather than us, flexing our muscle and saying, well, your 90 days are up, we're going alone, it seemed that would be rather foolhardy on our part, given the very good incentive that Fujifilm had to keep going. They were very, very keen, certainly have been in the media themselves promoting this product as one of the flagships of their regenerative medicine strategy. A clear commitment from them was evident, and so why not give them a little more time? Obviously, investors were concerned about that and felt that there maybe was some risk, but as we pointed out to those investors, if Fujifilm hadn't liked what they had seen in terms of the clinical study or anything else, for that matter, relating to the product or the technology, they could easily have just simply walked away and avoided the embarrassment or any issues associated with kind of dragging it out any longer. And, of course, that was not the case. They were very happy with what they had seen. They just needed a little more time. So, as I said, rather than cut off our nose to spite our face, we felt it appropriate to extend that period and to continue to work with them to complete the deal, which is exactly what's underway at the moment. We have an intensive engagement with Fujifilm and with our head licensor, WARF, the Wisconsin Alumni Research Foundation, to finalise all aspects of that deal and move forward with the Phase 2 program and beyond.
And when do you expect a decision now from Fuji?
Well, I wish I had my crystal ball out for answering that, but I have dealt with many different partners over my career, and I've learned that it's very foolhardy to try and estimate when the other side is going to complete their assessment or review or run through their management process to finalise a deal. And yes, you can put some hard dates in there, but it often doesn't pan out that way. So rather than commit to a date specifically, I point to the fact that we have given Fujifilm an extra 6 months, so that takes it out to September of this year. So we certainly hope that it'll occur, the close of the deal will occur before September, but I certainly won't be committing to a specific date at this stage.
Ross MacDonald, CEO of Cynata. Ross, thanks so much for your time.
That's a pleasure, and very, very nice to talk to you, Danny. Thank you for your time.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.